Hey everybody, welcome back. It's been a while since I posted a video, so here I've got another short knowledge drop for you, and it's on the topic of DNA transcription. Now, DNA transcription, why is this important for us to go into great detail about? As we start talking more and more about regenerative therapies, whether it's exosomes or Wharton's jelly, or talking about stem cells themselves, mesenchymal stem cells, and the agents, the compounds they secrete. We talked about cytokines earlier, but there's also growth factors. There's microRNAs. There's bioactive phospholipids that can regulate cellular processes to change the physical expression of a current cell, which is ultimately what we are trying to do with a biological therapy, right? We're trying to give a disrupted cell, a cell that's behaving badly, so to speak, and we want to change its behavior. And we do so with these various bioactive mediators that all of these, largely derive from mesenchymal stem cells, but they're also present in the matrix of Wharton's jelly, as well as in a free soluble form or bound to the membrane of exosomes. So DNA transcription is simply a level where this regulation, this objective of changing the phenotype of a cell to upregulate activity or downregulate other activities can take place. And this short presentation is going to show you how the process works, a lot of the key players involved, and what I want you to take away from this is not to memorize everything, okay? Hopefully you'll maintain a general understanding of transcription, what it means, but I want you to really focus on all of the different areas that can be regulated by cytokines and growth factors from a number of regenerative technologies that we're going to talk about going forward in much greater detail. So watch this short presentation. I'll see you on the other side. All right, everybody. Talking about transcription today, super fired up. I consider DNA to be the alphabet of life. You've heard me say probably that proteins are the language of life. Well, proteins are ultimately derived from our genetic code, our DNA. And therefore, I call DNA the alphabet of life. So again, this is an important process in cellular biology, but it's a very important modality to regulate expression of protein vis-a-vis expression of coding regions of DNA, also known as genes, okay? And when we talk about regenerative medicine, which I love talking about, it's all about changing phenotypes, getting cells to behave differently, getting cells to behave in a more normal fashion or more advantageous fashion. And one way is to control expression of various proteins at the nuclear level, at the level of DNA, at the level of transcription. And what you're going to see, I'm just going to hit the high notes here, but this is a very complicated process. But what I want you to get out of it as we move forward and I start talking about upregulation, downregulation in the nucleus, or regulating at the transcriptional level, or I want you to just have an immediate recognition, immediate recall of what that entails, and understanding the complexity of this process. So first of all, what is transcription? Transcription is the process. It's the means in which we take the message in the alphabet, right? That coding region of DNA, that gene, okay? And we turn it into an intermediate component called messenger RNA, okay? And ultimately, from that messenger RNA, we will get the proteins. So that's how cells influence behavior and influence their activity, is through the generation of various proteins and various mediators. But they all start with this transcription process, okay? Now, transcription is not the same as DNA synthesis, which occurs every time a cell divides, okay? When a cell divides, it has to replicate its entire genome, okay? And that's a different system. It's similar in certain respects, but it's a different system. And translation is a different system as well. Translation is converting the output of transcription, which is messenger RNA, converting that to a protein. And then that protein is going to go on to affect whatever cellular processes it has evolved to affect. Okay, so transcription, DNA to RNA. Translation, RNA to protein. Okay, so we're talking about transcription, DNA to RNA. So some of the key players you're going to hear me talk about, it's important to know. The first is probably the most important. That's RNA polymerase. This is an enzyme. It's a powerful, magnificent enzyme that has evolved to do so many different things. But what you need to know is this is the enzyme that binds to DNA, what we call the template strand of DNA, binds and then moves along that strand, and it catalyzes a reaction. Okay, we talked about the nucleic acid chemistry. We talked about phosphodiester bonds. It catalyzes that covalent linkage of nucleotide triphosphates, putting those nucleotides together in a string. Okay, and it's important that I say triphosphate, because what happens, how it gets its energy, these processes consume energy. Well, I should say they transfer energy. They cleave these phosphates, it releases energy, and that energy is converted into a covalent phosphodiester bond. So RNA polymerase, this is the main player that's reading that DNA. Remember, there's AGCT of DNA, right? So the RNA polymerase is reading that, and it's putting complementary nucleotides in place to match that code on the DNA. Now, the next two arrows, I'm just going to kind of combine them. Let's just call them transcription factors. These are components that activate, they can repress. The nomenclature is so varied for these compounds, and these compounds, which again are proteins, have a number of different functions, and a lot of them come together to enhance one another's activity or depress another's activity. But ultimately, you need these transcription factors to activate transcription. Okay, a lot of these factors coming together to stimulate the promoter, to stimulate the enhancer, which we'll learn about in a second, that will allow RNA polymerase to dock where it needs to dock and put it in a position to do its thing. Okay. Some more key players. I'm not going to talk about this that much, but there's another enzyme called helicase. Okay. And this is an enzyme, if you recall, our nucleic acids, our complementary base pairs, are held together by fairly weak hydrogen bonds. And if you remember, that DNA is wound pretty tightly in a double helix. Well, to get that RNA polymerase where it needs to be, we've got to separate those strands. We've got to break those hydrogen bonds. We've got to disrupt that double helix temporarily so that all of our transcription machinery can get where it needs to get, so that it can accurately and precisely read that DNA code. And helicase is an important enzyme in separating those DNA strands. Now, in bacteria, the bacterial RNA polymerase has that function. In mammals, in eukaryotic cells, it does not. It needs ancillary enzymes, ancillary proteins to disrupt those hydrogen bonds so that DNA can unwind at that important area so that RNA polymerase can find its home and start transcribing, start reading that template. An enhancer is a non-coding region of DNA, and it can be way far away from the start point. Okay, it can be way far away. But what happens when you unwind the DNA, it starts to loop. There's like this loop that forms. So it sets up this situation where you can have regions of DNA at great distances, 100,000 base pairs away, that can loop over and come into close proximity with other important regions from a great distance. Such as the promoter. The promoter is basically the activation site. We need to tickle this promoter, get that RNA to dock. It docks just downstream of the promoter for the most part. The promoter is a very popular area for a lot of transcription factor activities. You can have down-regulating transcription factors that block the promoter that will turn that gene off. But basically, when transcription factors are going to activate transcription, they're acting very near the promoter, okay? So here's kind of a busy diagram, but it shows you a lot of what I've been talking about to this point. You see how the DNA here is looped. After the helicase comes through, it creates a loop. And what happens, you can see the enhancer region, okay? And that's many base pairs away from the promoter, but the looping function brings those into relative proximity, okay? And then you see the promoter, okay? And then you see the RNA polymerase and approximately where that's docking right at the beginning of the coding section of the template strand of the DNA, which is the gene. And so this diagram shows a number of transcription factors docking at the enhancer and at the promoter, okay? So basically turning both of those sections of DNA, turning them on, so to speak. And by turning them on, that allows these mediator proteins. Sometimes this is just called mediator. It's this collection of like 30 proteins that come together in a hurry, and they transmit signals from the enhancer to the promoter and say, this is a go. The promoter kicks into gear, tells RNA polymerase, you're good to go. Then RNA polymerase will start its journey down the gene and start creating that messenger RNA transcript. Basically 4 stages to this whole process. You have initiation. That's kind of when that enhancer and that promoter come into contact via transcription factors and the mediator complex. You have promoter clearance. Once initiation happens, RNA polymerase is released from the promoter. It's released from its binding site of the promoter, and it starts its journey. Elongation, okay, is what they call pre-RNA. So that RNA polymerase is grabbing nucleoside triphosphates out of the area. It's catalyzing the reaction, putting them together in a complementary fashion to the DNA template and creating that messenger RNA. And then finally you have termination. And this, typically this happens, eventually the RNA polymerase at the end of the gene or the end of the coding region is going to reach what they call a polyadenylation site. So we're going to put a lot of A's in succession. And the RNA polymerase is pretty smart, and it says, okay, there's a lot of A's here. I must be done. It detaches. The pre-mRNA is released into the nucleus, into the nuclear cytosol, and then it's transferred out into the general cytosol. I think I did not use the right word there in the nucleus. Let's just say it transferred from the nuclear environment into the cytosolic environment, where it's eventually turned into a protein. But keep in mind, when a cell is trying to change something, change behavior, it's not creating one copy. So you've got multiple RNA polymerases docking, moving at the same time in succession like a ballet, creating all of these messenger RNA transcripts to create a volume of proteins to make changes within the cell. What if mistakes are made? RNA polymerase is more prone to mistakes than DNA polymerase. Generally, about every 10,000 base pairs, there's going to be a mistake that it makes. It connects two nucleotides, or it's reading the DNA wrong, grabs the wrong nucleotide, what have you. It has the ability to recognize this, and it can shift back, catalyze a cleavage, a removal of the incorrect nucleotide, and replacement with the correct nucleotide. And its fidelity at this is, amazingly sharp and precise. Because if mistakes are made, if there's a high volume of mistakes that end up in the final transcript, you're going to have a protein that may fold incorrectly, and structure is function with a protein. So if that mutation, if that incorrect base persists, you may have a problem with protein function, and hence being able to elicit the response that you're looking for. Now here's a video I'm going to play. Hopefully it plays. Oh man, it's not playing. Let's see if I can get it to play here. Sorry, folks. I guess it's not going to play for me. I'll put the link to this video in the notes. Go check it out. It's amazing. You're going to see all of the sections happening. It's only like maybe a couple minutes long, but it shows just that intricate ballet I talked about. You'll recognize how amazing this complexity and all of the things that need to align correctly for transcription to occur properly. I want to thank you guys for watching. I am going to put a link to the PDF of these slides in the description. Download those, use them at your discretion, and thank you so much for watching. All right, welcome back. I hope that was helpful. And again, I hope something you're going to take away from that presentation is the absolute marvel that is our biology. Something that complex that happens, I don't know, trillions of times over the course of days and weeks. I don't know. But all of those variables coming together in that ballet of protein to protein interaction, it just always amazes me. And as more research comes out, as more data emerges and our understanding increases, it's even more complicated. There's even more interactions that are taking place to synthesize protein, right? And I think I mentioned in that video, we're not just synthesizing one copy of a protein. This is happening dozens and dozens and dozens of places, one RNA polymerase right after another in a train, 20 to 30 nucleotides behind, just cranking out messenger RNA messages. And it's just a delicate balance in the way that a chemical molecule almost has intelligence to it, where it can recognize if it makes an incorrect base pairing. It can backtrack, cleave out, replace. It's unbelievable. But again, what I set up in the introduction to this video was that I wanted you to look at the complexity. And each one of those proteins is a potential regulation site when we're talking about changing phenotype of pathological cells, okay, or cells that are behaving badly. Interaction with those proteins, those transcription factors, disrupting the promoter site, or limiting access to the promoter or the enhancer regions, or interfering with the ability of the mediator complex to assemble and transmit those messages from transcription factor to the activation region of the gene so that it can be transcribed. Those are all ripe for regulation by the cytokines, by the growth factors, by microRNAs that we're delivering via exosome, via Wharton's jelly, or via secretions from mesenchymal stem cells themselves, okay? And that's what I want you to take away. The beauty of this process, but also the potential to regulate at the gene level. So as we move forward, and I use that type of terminology, regulation at the gene level, now you know what I'm going to be talking about. We're regulating at the transcriptional level. And a lot of those compounds we talked about in the exosome, in Wharton's jelly, in mesenchymal stem cells, they regulate at multiple levels. So it's just an incredible process. And I know it can get a little mundane and confusing, but just really appreciate the beauty of our biology. So I have put a link in the description. You can download this presentation as a PDF file. I don't think the video is going to replay, but I'll put a link to the video if you want to re-watch that. It's just something I connected to off of YouTube. If you haven't subscribed to my channel, please do. We took a little time off. I'm migrating a lot of my marketing activities to a new platform. That just took some time, and I haven't been able to produce, but we got a lot of good presentations in the pipeline. We're going to talk about telomere shortening. We're going to talk about protein translation or messenger RNA translation. We're going to talk about grapeseed extract. I've got some requests for that, so we're going to dive deep into that. And we're also going to keep moving forward with cellular senescence. And of course, exosomes, Wharton's jelly, and mesenchymal stem cells. So if you have any questions, leave them in the comments, or you can always email me at the email address below, workwithjeff@stayregenerative.com, and I'll do my best to get back to you. In the meantime, take care. We'll see you soon. Stay regenerative.